All right, Joni, you want to just give a brief introduction and we'll go from there.
Hi, welcome so much to North American Millets Alliance monthly webinar. We're really excited to have our guests here today. And we've got two incredible folks who are innovating with a lesser known crop, something that most people may not have had a ton of experience with. And I'm going to let Gary, I'm going to actually pass it to you to dig a little bit in. Gary's going to be moderating our panel today. And then I'd love for our guests to take a moment to introduce themselves as well.
Yes, I'm Gary Whitgriff. I will moderate. I'm from South Dakota, and I've been around millets for most of my life since we farmed millet as a kid, and I've worked with it and did international research and whatever. So I'm part of the North American Mill Alliance Board of Directors. Don Osborne and Joni is also on the board. In fact, they invited me into the board, so I'm thankful for that. So this is a millet. Finger millet is a millet that I'm not familiar with. So I'm going to be asking probably some questions just from, I'll just say from my ignorance. And as the only time that I've ever probably tasted finger millet was when I was in Uganda visiting our orphan girl. Okay, so we'll start with introductions. And Juan, you want to give a brief introduction, and then we'll get into the topic a little bit later. Okay, thank you.
Good morning, everyone. My name is Juan Salvo. I am a graduate student, a PhD student at Iowa State University. I'm originally from Argentina. I did my bachelor in agronomy in Argentina. And then I moved to Kentucky, where I did my master's degree in crop physiology. And after working a little bit with in plant breeding in soybeans in Argentina, I came back to Iowa State. In 2022, I started my master's in plant breeding, where I was introduced to the world of millets, and I started working with finger millet, proso millet, barnyard millet, and foxtail millet. And now I finished last year my master's degree, and I'm pursuing a PhD in a different topic, but in plant breeding in soybeans.
Well, thank you. All right, Shigiri, would you give your brief introduction, and then we'll go back to Juan with his presentation.
Sure. Hello, everyone. I'm Srikanth Panthulugiri. I'm a third-year PhD student at Iowa State University, majoring in plant breeding. So I'm originally from India. I have completed my bachelor's in agriculture. Then I have completed my master's in genetics and plant breeding. And during my master's, I worked in the legumes. But after completing my master's, I worked as a research fellow at ICRISAT, International Crop Research Institute for Semi-Arid Tropics. And I specifically worked on the finger millet crop, where we worked on screening for germplasm and making crosses and mutation, generating the mutation population in finger millet for an year. Then I moved to Iowa State, and here we are also working on soybean and millet projects combinedly. Thank you.
Thank you. We got a very interesting background group this morning. So certainly internationally represented. So Juan, why don't you go ahead and start, and we'll follow up with questions later. Please share your screen as you see fit. There we go. And expand it if you could.
Yeah.
Perfect.
Okay. Okay.
Yes, got it.
Okay. So as we were talking before, we will be presenting today on finger millet and the potential of this crop in North America. And we have 2 years of research that we have conducted here in Iowa. So I hope you enjoy it, and please ask questions at the end. So to start, this is a brief abstract of different news that were on newspapers and websites. What I want to show here is that climate change, it's a fact. We are having more erratic precipitations and drought events across the world. And then in addition to this, since COVID, there has been an increase in food insecurity, especially in the US. It reached a peak of 50 million people who had not access or couldn't afford getting nutritious food. So in addition to all these, here in Iowa, farmers are looking for a third crop to diversify their crop rotations. As you may know, in Iowa, most of the cropland is for soybeans and corn. So having a third crop that they can plant on a rotation with some winter legumes or cold-tolerant legume like a pea or mung bean, it's an interesting fact, and they are asking to try to find out if there's any other crop that we could use in the rotation. So what are millets? So as you are all part of this webinar, you may know something about millets, but it's a big, broad group of cereal species. Most of them are originally from different parts of Africa and Asia. And these species have been grown for centuries and have been part of the diet of people for centuries. In addition to this, these crops are very nutritious, have very nutritional value. And we, as I mentioned at the introduction, we have been working in 4 different species since 2022, finger millet, proso millet, barnyard, and foxtail. But for the purpose of the presentation, we'll focus just on finger millet. So my master thesis was focused on closing a critical gap. When we introduce a crop in a new area, we need to first perform a characterization. We don't know how it grows, how long it takes to maturity, what is going to be the yield if we will encounter some difficulties. In addition to this, knowing the growth and developmental stages of a crop are very important to determine when this crop is more susceptible to different stresses. And what we tried to do in my research was to figure out these 2 points here. So to start, I will talk a little bit about finger millet. Finger millet has an African origin. It's being the crop has its origin in Africa around 5,000 years ago. Later on was introduced in India, where it was widespread, and it's been growing in these environments for more than 5,000 years. It has acquired some tolerance to drought and salinity. In addition to this, finger millet has comprehensive nutrition. It's the highest cereal. It's the highest content cereal in calcium, iron, and zinc. And for us in the US, we can say that it's this crop is neglected in terms of research. There's very few articles talking about finger millet. Most of them are more for forage use or are more like a review articles. So we claim that we have done these characterizations. It's the first time in the US. So how do we see finger millets in Iowa in particularly? Well, we are not thinking in replacing corn and soybeans for farmers, but we see finger millets as an opportunity, as a crop that can grow in marginal lands where the water availability is scarce, where other crops do not perform well. But it can also be used as a catch crop when some crop fails, and then we can plant finger millet, or as a double cropping with a short season legume, as I stated before, some peas, excuse me, that are more cold tolerant can be grown first, the peas, and then later on plant millets in a rotation. Yeah, as I mentioned before, again, limited research has been done in the US. There's not studies talking about how well this crop can grow in the US. And there's a lack of adapted varieties to the US, and also a lack of understanding of the different stages of the crop. So here we will be talking today about 4 different projects or topics. The first one is, can we successfully grow finger millets in the Midwest? And for this, we performed study 2 years in 2022 and 2023. In 2022, we requested to USDA the whole finger millet germplasm collection. It was 692 different genotypes from all over the world. And we planted this in unreplicated trial here in Iowa. The unreplicated trials were mainly because the amount of seed that we received from USDA is limited. So it served for us as an initial characterization of more like qualitative traits, and also as a seed increase. So we collected seed color, head shapes, seed weight, leaf abundance, and lodging score on 61 plots of the 692 that were the plots that reached maturity and were mostly adapted to our conditions. Then in 2023, those 61 genotypes were grown in a replicated yield trial here in Iowa. And we measure plant height, days to flowering, seed weight, lodging score, and grain yield. In addition, we have a drought nursery location where we did some drought scores. So some of the results from this study, and I want just to address here that for these three traits, for yield, average height, and days to flowering, let me see if I can do the pointer. Yeah. For these traits, explain around 80% of the variation between our genotypes. But most interestingly, what we found here in Iowa is that yield and average height were negatively correlated, as well as yield and days to flowering. This is an indication that here in Iowa, in our environment, plants that are shorter yield more, and plants that flower earlier yield more. So this is something interesting, because when we perform selections, we can do some sort of indirect selection for shorter plants and earlier flowering that potentially will have a higher yielding. And then I will show you just 5 of these 61 accessions that we have. So remember, this was done only 1 year, 2 replications. So I know these yields may be kind of high, but just we need to perform more research. But we see here that there are some accessions yielding around 5 tons per hectare. This is very high. All the literature that we found in different places of the world is reporting a maximum of 3.5 tons. So this is above that threshold. And honestly, we didn't do any extensive fertilization or irrigation. So it was in a rain-fed environment, just 1 nitrogen application. And you can see that we have high yielding, and then average drought score. So score of 1 indicates drought tolerance. Score of 5 indicates susceptibility to drought. Again, this was done only in 1 environment, but we see that we have high yielding varieties that seems to be drought tolerant. And then Srikanth will probably later talk about, we are using these lines, some of these lines, to perform crosses and develop populations in our breeding program. So in conclusion of this first part, we observed that there is a large variation among these genotypes. We identify high yielding accessions higher than 3.8 tons per hectare, and also some accessions that were drought tolerant. We observed that short plant stature and early flowering was translated in higher yields, but still we need more research, more years, environments, and replications for this study. What I want to mention is that this first part, it's going to be published. We send the paper to a journal, but the most important is that all the data that we generated will be publicly available for other researchers to use. So all the raw data of the 61 accessions that we evaluated, it's going to be an Excel format, and it will be available for download and to work with it.
All right. Well, go ahead.
Yeah.
Juan, go ahead, finish up.
Yeah. So, and the second project that I work in my master was the growth and developmental stages in finger millet. So, many of you may be familiar with growth and developmental stages and how these charts are basically supplemented with images and figures and photos. So what we tried to do here was to use the BBCH scale. BBCH scale is a standardized phenological scale for all species that potentially can contain up to 100 different stages. So it's a very extensive and can be somehow complicated for farmers or even researchers to understand. So what we tried to do was to adapt the BBCH scale to a more user-friendly scale, and then supplemented or complemented not with just photos and images, but with 3D reconstructions of the plant that are embedded in a virtual reality environment. So I will show you this briefly. So in 2023 and 2024, we conducted greenhouse experiments with 2 genotypes of finger millet, and we recorded phenological stages and took photos on 2023 and 2024. Additionally, in 2024, using iPhone 14 Pro equipped with a LiDAR camera, we did a 3D scan of these plants in a rotation table. You can see in the picture, we put the phone and we have a 360 image around the plant. And then with one software and the help of students from mechanical engineering here in Iowa State, we were able to reconstruct a 3D plant of millet. So I will show you first, this is regular images that we took, and you can see in the top part, these are vegetative stages, leaf development, tillering, and head emergence. And in the bottom part, there's a flowering and seed ripening and maturity. So in our description of the stages, we have the text describing the stage, the image, but also the 3D reconstruction. So the BBCH scale, you have in the first column, the proposed scale, it's what we adapt from that scale, and then the description. So you can see that we use approach similar to what it's in corn or soybeans, where we call B to vegetative stages, mostly leaf development. And then H stages to what it's heading. So we call B1 to BN, as many leaves are expanded on the main stem. H, it's when the head start to emerge from the flag leaf. And we have reproductive stages with R describing different stages of the reproductive stages. This will be also in a publication already submitted, so hopefully will be available soon to all of you. But then to finalize my part, I want to show you the 3D models in the immersive environment. I hope the video runs well. This is a collaboration with Professor Krishnamurthy and Shambhavi Shoshi from mechanical engineering. And as I mentioned before, this is in order to complement these developmental stages, we create these virtual realities, sort of a video game where the user can have a headset and some controls and can move inside the environment. Right now, we have just the video because it's the way we can do it. But in the future, we hope that you can enter this immersive environment, navigate from your computer. So let me see. There we go. So basically, this is what the user is watching in the headset, and we have different phenological stages where you have the plant, and then you have a description of what is that stage. This can be used for farmers, researchers. It's sort of a training method. Like when you have someone new working in the crop, you can first train the person with the immersive environment and then let them do the ratings in the field. So as long as we know it's the first time these have been done this way for developmental stages. And I know some of the quality of the plants it's a little bit finicky, but we're working on that. And if you are using the headset, it's much better than probably what you are seeing with the computer. So that's all my part, and now Srikanth will talk more about the crossing techniques in finger millets.
Well, thank you, Juan. This is, I have probably a dozen questions just to ask you alone. So let's move on to Srikanth right away and give your presentation, because questions are going to be loading up. So great presentation. Thank you.
Yeah, thanks.
So, thank you, Juan, for presenting your work. Today I'm going to present my work on finger millet in the different area, that is initiation of the breeding program and generating the genetic, creating the genetic variation in the finger millet. The first part, can you go? Okay.
That's Juan. I think you're still sharing your screen. There you go. Okay, sure.
Yep.
Go ahead.
So after the characterization work that was done by Juan, we have identified the potential candidate lines from in finger millet, and using those lines, we have used 8 different cross combinations to make crossings between the male and female. For this purpose, since finger millet is a predominantly a self-pollinated crop and it has a clistogamous flowering condition, so there is a need. It is very hard to make manual emasculation. So there is a need to make artificial emasculations using different techniques. And based on the literature, we found cotton ball method, gibberellic acid treatment, and hot water treatment has worked in the past. But cotton ball method was predominantly used in the Proso millet, but for the first time we are using in the finger millet here at Iowa State. And out of 3 emasculation techniques, we are able to make crossings and crossing attempts in the cotton ball method and gibberellic acid method. But due to the synchrony issues, we are not able to make any crosses in the hot water treatment. So in the first method, in cotton ball method, we are using a cotton ball which is dipped in water, and after chopping up the flowering portion and after removing the excess fingers, this wet cotton ball is placed on the bottom in the polythene bag, and these plants are exposed to high temperatures. For the high temperatures, we have used 30 Celsius temperature in the growth chamber, and also 72% relative humidity. So once these plants are exposed to high temperatures and high humidity, on the next day, the anthers used to come out, and they don't decize, but they stay on the top. So on the next day, we can remove those anthers with a forceps. Later on, we can bring a male parent to club with the female, and in that process, we can make a bagging. And this after successful pollination, we can see the seed set on female panicle. In the gibberellic acid method, we have used gibberellic acid is a growth hormone. However, when it gives in the higher dose, it can act as a emasculant. So for this purpose, we have used only one dose of 1500 ppm, and this is our first attempt. So we restricted the one dose, and this gibberellic acid treatment has to be conducted in 2 consecutive days. That means on the day 1, if we perform the day 1 dose, and in the day 2, we have to give the dose 2. And if there is any gap, if there is any gap between these 2 days, then as it is a growth hormone, the panicle will emerge very quickly, and it will show. We may lose the crossing emasculation thing. So after after gibberellic acid treatment, the panicle will show the sterile spikes coming out. Once if you see the sterile, and if you can club with the male parent, and if there is a successful crossing, you will see the seed set on the female panicle. And next. And during spring 2024, we have started this crossing between the finger millet. Out of 8 cross combinations, we found seed on 6 cross combinations in using cotton ball method. In gibberellic acid method, since we have used the higher dose, and these lines are they show high burn symptoms on the panicle, and after clubbing with the male, we didn't see any seed set, but we are succeeded at getting seed in cotton ball method. And you can see in the images, the seed set were like we observed very low seed set from 2 to 49 seed set, and it also depends upon the number of attempts of crosses that you make. So if you have a more cross attempts, you may get more seed, but out of in cotton ball method we have performed 24 emasculations, and out of 24 we have saw the seed set in 12, that is 50%. But in gibberellic acid method we have emasculated 15 plants, and after clubbing with the male, we didn't see any seed set with the gibberellic acid, but we have seen the deformation and burn symptoms of the female panicle. Yeah. So this is about crossing, and the other project that we are working on is how to bring the genetic variation in the finger millet. And for this, we have used the mutation breeding last year. And yeah, Juan, go ahead. Yeah. So we have used 2 finger millet genotypes, and which were treated with the 500 GY and 600 GY doses using gamma rays. And we have seen some chlorophyll mutants in the field, but as part of the field management, it was affected with the herbicide, and we couldn't harvest any millet mutants in the F1 M1 finger millet mutants. And we also use the mutation breeding in Prof millet just for information. And yeah. So these are the few chlorophyll mutants that we observed in the field in the summer 2024, and we are planning to continue this mutation breeding in this summer as well. So from conclusion, from the last 2 years of finger millet study at Iowa State, we found that finger millet is suitable for production in Midwest region, and we have seen a promising results. But however, as Juan said, we need more data to get a better conclusions. And we have started our variety development program, and also we are creating genetic variation through mutation as well. And the future work includes we are planning to evaluate the F2 populations that we have increased in the last in the growth chamber and we are also field evaluation of the F2 populations in the field, and we are targeting to select the desirable segregants from the F2s. And the other work that we are targeting is to continue the mutation breeding in the finger millet again in this summer. Thank you. And if you have any questions.
Yes. Why don't I just start with you? I think everybody is curious, and this is obviously down the road, but when do you think you will have varieties adapted for Iowa available for farmers?
So right now we are evaluating the F2s in this summer. So once if we are able to get a good lines, probably we may get a line within 5 to 6 years probably. Okay.
And so you can generate what, 2 generations a year or 3 generations a year in greenhouse in Iowa? And
if we can send the seed to winter nursery, we can get it 2 generations per year. Okay. But as far as in Iowa, then since in F3s and F4 we will have the more seed, so it may not be possible to grow in the greenhouse or growth chambers. But in the winter nursery, we can get 1 more generation. Okay, before I jump to Juan, and from a breeding standpoint, how many days are you projecting to maturity? You talk early varieties, as did Juan, but how many days from planting to physiological or harvest maturity? So in finger millet, generally the early maturities are like from 90 days to 110 days. But since we are targeting for maturity and yield, and if we can reduce at least 10 days, then it will be really great. So the crossing design itself, we are targeting for early maturity, high yield, and with lower plant height.
Okay, good, good. Juan, I'm going to go with you for a couple of questions,
Yep.
and we can. You're moving finger millet from basically Africa, which is plus or minus the equator, and into Iowa, and getting basically 10% of the varieties that were offered by USDA into physiological maturity, to harvestable maturity. I think that's amazing. So are the plants quite photosensitive, and are they sensitive to Iowa humidity compared to maybe some of the droughty areas of Africa or India?
Yeah, great question, Gary. Yeah, so we found out that finger millet is a short day plant. So we found out that the varieties that we were able to harvest may have some insensitivity to photoperiod. There are some reports that there are varieties insensitive to photoperiod, and one of the things that we encountered was that the 2 years that we have these experiments, we were planting later than what we would like to do. So we planted in June here in Iowa, mid-June. Ideally, we want to plant earlier and see if we can reduce the time to flowering. But yeah, we were able to help like 10% of that genotypic variation. As you can imagine, in those almost 700 accessions, we saw a bunch of different things: super tall plants, not flowering, some very early flowering. So it was a wide selection. And I assume that if a different breeding program will do it in a southern latitude, like, I don't know, Mississippi or someplace southern, they will get different accessions. About the moisture, I'm not sure about that. We didn't see any issues in the heads. I don't know.
Could you answer that as far as what you see in the greenhouse versus in the field reaction to humidity? When you have hot, sunny days, is that preferential, or do you see diseases, for example, developing when you have too much humidity?
Well, in the field, we didn't see any issues. Like we had rains in the summertime and high temperatures here in the summer. We can have up to 100 Fahrenheit, and we didn't see any disease. We didn't see any pests at all. So that's something striking. And again, it's been only 2 seasons that we have these experiments, but that's one of the things that we read about finger millet, that it's very resilient and it doesn't have any many issues with diseases or insects here in Iowa. We know that there is some blast that affects mostly the head of finger millet, and that's a big issue in Africa. But so far here in Iowa, it's not a concern. I know that this disease, it's a similar strain of what is in rice. So I assume that if we move this crop to southern locations, southern environments where rice, it's a crop production area, then there might be some issues. But
okay,
yeah.
All right, Srikanth, what are you seeing from the greenhouse versus the field? What are the differences that you're seeing?
In the growth chambers, and we grown the plants in the growth chamber. In the growth chamber, the plants grows since in the field the height of the plant is a little bit lower as compared to the growth chambers. In growth chambers, maybe because of the lights, the plants elongates a lot. So it makes when we are doing the crossings and we have to bring out the plants from the growth chamber, and we have to match those height differences between the male and female. And that is the one problem that we face in growth chambers.
Okay, so that means you brought that up. What about lodging? You obviously are breeding to prevent lodging, and that's one of the critical things when you're talking. I can't believe the yields that Juan showed. That's unbelievable yields.
Right.
If that is even 3/4 realistic potential yields in Iowa fields, I suspect you're going to have terrible lodging problems.
Yeah, and I have some pictures that we took with the drone, and we have strong winds here in Iowa also in the summer, and we have severe lodging. So we took those ratings. Some of the plants lodge, but then come back again, stand up. But there were some instances where some plots were lost because the combine couldn't go that low to pick it up. So that's one of the reasons why I think we saw that correlation between lower height and high yielding. That could be probably because the taller plants lodge, and then there was some yield reduction there because of losing seed or shattering. But one of the big things that we are struggling and we need to work on is to synchronize the maturity between the head and the stem part. Right now, we are having some instances where the head it's mature, the seeds are getting dry, but the plant is still green and has a lot of foliage. So the combine it's struggling to harvest that. We need to put the head of the combine very tall, try to avoid introducing green matter inside the combine. So trying to breed for that synchronization, or we can probably use some desiccation treatments, but we haven't get into that part.
Okay, we got an excellent question coming in, and so just my experience out here with here in Dakotas with wheat and millet and other things, there is ability
Gary,
to
I
use,
have some suggestion to both of these speakers.
Good. Using stripper heads, I guess that's what I was going. Please, what's your question or
My
suggestions?
Question is, you are introducing finger millet, particularly in Ohio, where the corn, belt corn, and soybean is the main crop rotation.
Yes, Iowa. Yes, okay.
And Iowa, there is a problem coming of cyst nematodes. And to overcome that, when you are getting the variation in various type of accessions and you are observing the plant height, maturity, cold tolerance because of this all system what the Ohio environment is. In that one, you should also focus on its root system. How deep the root goes down in the soil, and if this finger millet is doing like pearl millet, then there is a solution for the cyst nematode, which is coming very highly. We already have in Ontario. It has moved to Michigan, and Ohio is not very far.
Right. So cyst nematode are breeders, Shrith or Juan, are you testing for cyst nematode?
The breeder can be involved with the nematologist, and if the root system behavior are deeper, going to 4 or 5 feet, then it can be a very good grain crop as well as a rotation crop in between soybean and finger millet.
Yeah, good.
Like
good.
We are doing here, like a forage pearl millet, green pearl millet, and sweet pearl millet as a rotation to potato and tobacco and strawberry, where we have Prettylancus is the main nematode, Mellides and Prettylancus.
Well, thank you, Om. Let's go back to our speakers. Can you address the
nematode? We have also that pearl millet is working, but if the finger millet can do very good work in Ohio, it will be a good achievement of our research.
Fantastic comments. Any testing on cyst nematode, both in the greenhouse and in the field?
No, we haven't. We are part of a bigger breeding program that we work with soybeans too. In soybeans, we have the phenotyping experience for cyst nematode, so it shouldn't be like a big change. We could probably do some testing for cyst nematode. We have some areas here in Iowa where we can test for that.
Well, you
but
have some extreme areas in Iowa with
Yeah.
this nematode. Yeah, absolutely.
But about the root system, one of the strategies of finger millet to avoid drought, it's a deeper root system. So
Mhm.
I think, and our lab has some experience or have bunch of experience in treating with root phenotyping. So
Okay.
that's something that definitely we could study. Yeah.
Okay, good. We have a
Thanks.
question
Thanks.
coming. Thank you. Thank you, Juan. We have a question coming in about from USDA about sprouting in the field. Do you see with the high humidities and rains towards harvest that the seeds actually sprout? Let's go with Sharik on this. Seeds actually sprouting in the head, either in the greenhouse or in the field.
In the greenhouse, we didn't. In the greenhouse, we didn't see any spotting in the finger millet. But in the field, during my experience with the mutation, we didn't saw any those kind of spotting. But in the characterization, maybe in some genotypes, one may have observed. But the lines that we selected, probably we didn't observe those spotting issues.
Okay.
Yeah.
All right.
No, not too many. No.
All right.
Not in finger millet. Yeah.
Gary, I have another
Yes, go
clarification
ahead.
from Srikanth. That how about the settling in this finger millet? Seed settling before combining.
So we have one slide showing like without crossing, there is a large amount of seed set has happened in the greenhouses growth chamber. But after during emasculation, we will remove the excess spikelets and we will only keep the targeted spikelets that we expect to cross and we will get the seed. But after cross in GA3 treatment, we didn't saw any of those seed set, but in cotton ball method we have seen, but not in all, but we found very less seed per finger. And the lowest seed we have seen is 2 seed per finger, and the highest seed that we have seen is like 49 seed per finger.
Wow. Okay.
Okay.
Wow, that's interesting. Very
For
interesting.
example, if you have grown in the field, and I think what is name Juan can explain me agronomy. How much percentage of moisture are there when you are combining it?
Good question.
Yeah, well, it depends, but we got some. So our combines, we cannot measure moisture on these combines because there's no calibration course for this crop. But I can tell you that we harvest pretty wet the first year. We have to take it to the dryers, dry to almost 0% moisture, and then correct to 13%. But it depends on the variety. There were some that were very green and some that were very dry. If I have to say, there probably be some of these genotypes were harvested with around 30% moisture, at least.
Okay, that's way above what farmers would even consider drying. So because we're talking about farmers, and this is for both of you researchers, obviously you're in a rotation or expect to be in a rotation, whether it be a catch crop or rotation in Iowa. And I'm an agronomist for many years. I don't know of a herbicide in the US that would allow finger millet into the rotation. Can you even come back the following year with finger millet after corn or soybean herbicides? And if so, if you're not, are you testing them?
Yeah, so weed management is one of the biggest things that we observe working with this crop. So initially we started with a 15-inch row spacing and we moved to 7.5 inch space between rows, and that helped a little bit. But weed management, it's a big issue. We did some research with a seed protectant, the seed protectant that is used in sorghum. So I think it's called
like concept treated. Okay.
Yeah. So we try with concept and we run some experiments for proso and for finger millet. For proso, it worked well. For finger millet, it didn't work well. In fact, last year we have a huge damage and low emergence. But we are interested in trying to find out some seed treatment or some herbicide that will help us to control weeds, because even if for us in breeding with such a small plots, it's very complicated to keep them clean and try to manage that.
Absolutely.
Well, I can advise you something. The work we have done in Canada, particularly in pearl millet, we have done all the studies related to rotation with soybean and corn. And we had a herbicide trials conducted by provincial government, and they come up with very good herbicide to control the weeds, particularly the broadleaves as well as the grass weed. And we are using prowl and partner combination.
Okay.
After we cannot apply pre-emergence. We have to apply after the plant germinated and have 2 to 4 leaf stage.
Okay.
It may be the same case with your finger millet and proso millet, but you have to do the herbicide trials, particularly where you have soybean or where you have corn, and follow that land, and it should be done very carefully so that in future any genotype you are developing, particularly Srikanth, that will be very helpful to become as a farmer crops. Thank you.
Absolutely. Very, very good point. We got a
That's a
question
great
coming in.
suggestion.
Please. Oh, go ahead. Answer.
I mean, that's a great suggestion.
Yes. And this is when Olm has worked in and was on our program last month and has worked with pearl millet for many, many years, and so understands both the agronomy as well as the breeding. But we have a question coming in. Would you go ahead and speak? I can't see who it is.
I think it's Krishna Khan. Yeah, go ahead.
Yeah, this is Krishna. I'm from Delaware State University, and I primarily work on horticulture crops, but I'm interested in this crop. So thank you, Srikanth, for inviting me for this wonderful presentation.
All right,
So
great.
I
Go
have a
ahead.
couple of questions. First one is,
Please.
you said like you are using mutants. So I know that these mutants, they will be unstable for a couple of generations initially. So for the Iowa State, when do you think they will be stable to be used for like a variety trials and then released as a variety, like if you are interested in that? And my second question is, are you using like a speed breeding to get the more generations so that you go quickly to for expediting the variety release? So these are my two questions to you.
Yep. Thank you, Dr. Krishnan. The first question regarding the mutation. So generally we see a lot of segregation, mutant segregation in the M2 generation. Once we do the selections in the M2, then we will do a early yield trials like in M3 and M4, and we keep selecting the best mutants until M4. From M5 we can go for yield trials, and the lines which are performing well can be forwarded for advanced yield trials, which can be released as a variety in the future. And for the second question regarding the speed breeding, we till now we didn't use any speed breeding for generation advancement, but we have increased the seed in growth chambers. For we got the less seed in F1s, and we have increased those seeds in the growth chamber. But probably in the future we may think if the to grow as in the speed breeding thing.
All right. Thank you. Thank you.
Thank you.
Follow up.
Thank you.
A question came in on minimum soil temperatures. What are the soil temperatures you can go with germination? Basically, what the question I think is asking, when can they plant it in Iowa? If corn is planted in April, beginning in April, and soybeans say the end of April, 1st of May, when can you expect finger millet to be planted?
Yeah, I'm not sure about the number for that one, but there are some of the genotypes that have been grown in the high mountains in Nepal or in different places in India where the temperature is kind of low. So I assume, and I've done just a short experiment germination in the chamber with 4 Celsius. and I was able to get seed that was not damaged and germinated. As I was said before, we planted in June here in Iowa, so probably that's too late. I would like to advance and plant earlier, but I would say I wouldn't go lower than 10 Celsius, like what we do with corn. I wouldn't plant much earlier, because once the plants emerge, if there's a frost, it's going to die.
Yep.
So, but yeah, I don't have a specific number. We need to do
Okay,
some.
I will reply that question to you people. We are northern to you, all over Canada, and we are growing the pearl millet ranging from 43° north latitude to 52° north or 56° north latitude. And in corn, our recommendation is you plant the corn when the soil temperature is 12 degrees Celsius. Now you convert in your Fahrenheit. We use Celsius here.
Yep.
And that is the best. We have tested all these corn, soybean, all sorghum, pearl millet, foxtail millet, and we have no problem in its germination and any frost effect coming out of it.
Okay, thank you. We got more questions coming in. This is fantastic. Let's move to the actual demand. Melanie from USDA has a question about US demand. Where do you see demand for finger millet? And is that demand for grain? Is that demand, do you see as a prospect for forage? Obviously, we're talking grain today, so let's keep it focused on grain.
Danny, do you want to take that one, or?
You can start, Juan. I can come in if needed.
Yeah, so the focus of our program is for human consumption. Finger millet is for human consumption. Proso millet will be more as a forage crop. So we are working on, and our program works on conventional breeding. We don't do GMOs. So we are targeting, food industries. There's a bunch of different processed foods like beverage, flour, cookies, snacks that can be done with finger millet. So that's our main focus right now. It's for human consumption, but could be used as a dual purpose also. It's a crop that can be used for several uses. Yeah.
Yes, great. And so do you see it as obviously most grains in Iowa, or the acres of corn, virtually goes all to feed or ethanol in very limited amounts to actual food consumption. Do you see that as a market for finger millet, that it would go into the feed market? Because if you have large quantities, the food millet may not be developed at that point, and food processors need access to large supplies and quality seed, quality grain. So can you address, one of you address the grain standards, maybe Danny could address that, the grain standards around that, and how you see that market developing?
Yeah, so Gary, thanks for this, and I apologize, I'm delayed.
No, please, I'm glad you're on. Thank you.
Yeah, no, thank you. So Gary, one of the things, I'm going to make some, more philosophical comments and try to answer.
Please.
Like, look at the number of people that are on the call, like the community of millet. It's very small. And what we are attempting to do here is that we are thinking of historically, some of the cultures, they had millet as primary food source for the carbohydrate and some of the protein that came in, and minerals before they moved to a wheat and other type of diet. There are these crops, they have a lot of benefit, which I'm sure that Juan and Srikanth, they've talked about. So our take on this is that when a new crop comes in, it's going to take a lot of different stakeholders to come in and standardize things. If we think of food and feed, it makes it a little bit more complicated for us to do that breeding, because then we start adding more traits and different considerations. So we decided to start on the food, because that's what finger millet is primarily used in some of those healthy food alternatives here in the US or North America. But then, aside of the trade issues and all that, if we didn't have those issues with tariff or other things that might come in play, there are emerging economies that probably may have need for US to provide that type of grain. So we meet the need of that healthy market, whether it comes in those snacks or breakfast cereals and those bars and all that, or just as porridge or whatever it may be. So we are not thinking of a feed for that reason, because there might be other crops that might be more suitable for it. It does not mean that we may not go there, because as we start doing some of those nutrition studies and some of the other animal science related studies, we may find out that there are strains or accessions that are really good. So grain standards have, as far as I know, we have not looked at them in detail the way probably we need to, because we are very early stage, as probably Srikanth talked about. We are just getting into those breeding families. But that's how I would, respond to the question, Gary and Melanie, that you asked. It's that it will need a lot of us, which I appreciate you folks, what you're doing. You're raising awareness, and we need to expand so that people know. I've already received, so I wrote a grant a couple of years ago on this. It didn't get funded, but that grant was really good because I made connections with different companies that are working with finger millet. And I've had conversations with some farmers that are interested in growing and are growing, and some companies that are getting finger millet. So there seems to be that, at least at some level, there is an interest in this as a food crop, that they want to source seed.
Oh, wow. That's fantastic. And I like your presentation in that you're very specific. You're not going off to wherever they can use the stuff. You're going to, and in the case of finger millet, you're going to a specific market. And so that's fantastic, Dr. Singh. We have, well, let me go to Sharif. Why don't you bring up and address the quality standards? If you're going into a food market, what are you looking at as far as characteristics in the seed that you're trying to produce, in the grain that you're trying to produce?
Yeah, still we are quite early in the process, so we have not done those nutritional profiles. We are relying on what the literature reports on different nutrients, so that the macro in terms of just thinking of protein and the type of carbohydrate, with some of the protein properties of the gluten and all that. So it's really healthy for a lot of people that might have inability to digest it and some sensitivity to it. But then, things about calcium, some of the other minerals that it comes at a very high rate. So those are some of the traits that we will be looking at, that when our seed comes in the market, and those people, as Don, you put in your chat, those people that are trying to source these millets, we can show them that the new varieties that are coming, they have an advantage to the current varieties. No one's going to say that I don't want more calcium in my grain, because
Right.
that just comes, it's sort of Mother Nature's way of freely giving us something with the beautiful crop that I find finger millet to be. So we have not done those assays ourselves, because our population, in the breeding population, are not at that stage. But some of the characterization that we have used are through collaborators and the literature review. We do have a very robust food science program here at Iowa State University, and those collaborations, those discussions are happening. So anyone who has an interest in this space, I encourage them to please reach out. It will take a community for this to get in the hands of people. Why should people not have access to healthy grains? The grains that be grown with less resources.
Yep, absolutely. And I like the question. Let's
Gary,
go with.
if I can add to Dr. Singh.
Yes, please.
In the history of this whole millet program, or millet, whatever you have, six, seven type of millet, it has already been identified. They are free from gluten, number one. They are rich in high minerals. They are also well adapted now by, if you look toward the food processing system of Asia and then Africa, 100% there is a market for this if it is developed in North America. And within North America, there is a lot of demand. For example, sorghum and millet are the main source of pet food. All these I think we have more consumption in pet food of our crops like millet and sorghum, if you look into dog and cat and birds, this is the biggest market if we look into. And
Yes.
data is already available on USDA and other departments. How much is the consumption we are doing of sorghum particularly, which are the second or third largest production in the world. Thank you.
Yeah, no, good point.
I appreciate that. We are coming up to the hour, so I would like Dr. Singh, you're on. Why don't you summarize what you want us? And we have one more question come in about rain-fed foods in Canada has a millet for milk. Obviously, that's a food. Why don't we address that in your summaries? I want each speaker to summarize here in the next couple minutes.
Yeah, I'll start. I know I was late, but I'll summarize with this, that we have to think, because different millets, they might have their own unique advantages. We have to think it through, that we create the millet in a way that there is a market and very like almost like branding that is associated with different millets, so they're not competing against each other. I think we have to get consumers the ability to source these types of millets for the unique opportunities. I really like the example of pet food. There are studies that show that pet owners, they invest a lot more of resources, even for medical expenses and food for their pets, than they might not even for themselves. It is an incredible market. So if there's some advantages to be gained there, I think that could be another way to think about whether it's finger millet or any other type of millet. It will require a community. I'll do this one challenge for all of you. Agronomic production of finger millet it's complicated because we do not have good herbicide chemistries to do weed control. So there are lots of things that still need to be resolved in order for us to get this, in the North American type of cultivation. So in some of the other countries, they are able to cultivate with a lot more manual labor, and that's probably not going to be the case here. So we have to have that continuum all the way from agronomic to the final, value added that goes in the product. So that's how I would summarize that. We are doing research, we are starting it, but we want partnership and collaborations with all of those people that are interested. It's not going to be one or two groups cannot make it.
Very, very good points. Okay, let's go summarize from the breeding standpoint. Srikanth, why don't you come in and tell us what you see from a breeding standpoint that are main points that we meaning the recording, we're going to be on YouTube, so keep in mind that this can be shown throughout the world. So from a breeding standpoint, go ahead and tell us what you want to summarize.
So from breeding point of view, like in finger millet is a highly self-pollinated, so there is a need to standardize a protocol for emasculation. So till now, like from literature, we didn't find any reliable emasculation technique. So if we can standardize that, then we can keep making crosses, and we can make more breeding populations in the future.
All right, and you did, just briefly, you did bring up the seed production ability when you cross. Obviously, if you can get only one or two seeds in a head, that's not very good. So you're selecting, isn't that a critical part of your observations, testing?
Sometimes like we may end up with like getting like only a few seeds, but it depends upon how many crosses you are making for each cross combination. There we can get the more seed per each cross.
Okay. All right. Thank you. And to wrap up, Juan, again, we encourage everybody on the audience to send your friends and family and fellow researchers to YouTube within a few days. This will be posted and permanently available on our Millets Alliance YouTube page. So just enter Millet Alliance and you'll see the various hour-long YouTube series, as well as this one, of course, is going to be the last one. So Juan, from your standpoint, please summarize what you want the audience to hear.
Okay, thank you, Gary. Yeah, what I can say is that when we started a breeding program from the scratch, this crop has not been grown extensively or characterized in the United States. We find encouraging varieties that yield wood here in Iowa. And then we are super excited of keep doing it, keep pushing and trying to develop varieties that are more adapted and more more adapted to Iowa environments and Midwestern environments. And hopefully, if we did all this work in 2 years, we are excited of more to come. So, yep.
Okay. All right. Thank you so much. This is an amazing webinar. You bring out, all three of you bring out things that that every grower should be looking at as far as alternative crops, because they don't want to produce a monocrop, corn on corn. They don't want to necessarily plant just corn and soybeans. They want something in the rotation and something that can bring value outside of just a standard, local elevator takes what I have. And so thank you for your breeding efforts. Any other last comments before Don puts up the final departing slide?
Thanks for the invite. Really appreciate it.
All right. You got an amazing team of researchers, Dr. Singh.
Dr. Singh, I would like to collaborate with you people, and I can give my about 45 years experience of breeding in Africa, Asia, and North America.
Thank you.
Perfect.
Thank you.
Perfect. And along with that, if what he's referring to is a webinar that he was the main speaker last month on pearl millet. So I'd advise you to review that pearl millet video, because there's excellent information, just like you presented this year from a Canadian standpoint. Okay.
Thank you.
Thank you.
All right. With that, we're North American Millets Alliance. Board of Directors recommends that you go to our website. You take in monthly webinars. We have them the third Thursday of each month at the same time. So please, join us, and we look forward to basically spreading the word on what we can help researchers, breeders, farmers, and the marketing system do and food system, obviously, to bring more millets into production in the U.S. and bring utilization of those millets. So thank you very much for joining this webinar, and I really appreciate all your insightful questions and certainly presentations. It's very, very good presentation today. So thank you all.
Thank you.
Thank you, Eddie.
I appreciate it, Juan.